In this video, we are going to learn about the evolution of the aerosol preparations. Here, aerosols are the pressurized packages, and many tests are necessary to ensure the proper performance of the package and the safety during the usage at the same time storage. Here, pharmaceutical aerosols can be evaluated by series of physical, chemical, and biological tests, which mainly includes flammability and combustibility test. Under this, we are going to determine flash point and flame extinction test. And physiochemical characteristics can be determined by conducting various tests on vapor pressure, density, moisture content, identification of the propellant, concentrate propellant ratio. These are all needed to be evaluated. And performance tests like aerosol valve discharge rate, spray pattern, dosage with metered valves, net contents, foam stability, particle size determination, and leakage test are needed to be conducted. Some biological characteristics are also needed to be evaluated for the aerosol preparations. So we will see one by one here. The first test is the flash point determination. So this is determined by use of the standard tag open cup apparatus. The aerosol product is chilled to a temperature of about 25 degrees Fahrenheit and transferred to the test apparatus. The test liquid is allowed to increase slowly in the temperature, and the temperature at which the vapor ignite is taken as the flash point. So although the test is still used, the results are limited value because the flash point obtained is usually the flash point of the most flammable components, which in the case of topical pharmaceuticals is the hydrocarbon propellant. And next test is the flame projection test. So in this, the test indicates the effect of an aerosol formulation on the extinction of an open flame. The product is sprayed for about 4 seconds into a flame. Depending on the nature of the formulation, the flame is extended, the exact length being measured with a ruler. Okay, so it is nothing but just spraying the aerosol product over the flame and then measure the length of the extended flame by simply using a ruler. So this is a manual method. And next one is the vapor pressure. The pressure can be measured simply with a pressure gauge or elaborately through use of a water bath, test gauge, and a special equipment. So it is important that the pressure variation from container to container be determined, since excessive variation indicates the presence of air in the headspace here. A can puncturing device is available for accurately measuring the vapor pressure. So methods are available for aerosol package in both metal and glass containers here. And next test is the density determination test. Here the density of an aerosol system may be accurately determined through the use of a hydrometer or a pycnometer. So these methods, which have been used for the density of non-aerosols, have been modified to accommodate the liquefied gas preparations. A pressure tube is fitted with a metal flanges and a hook valve, which allows for the introduction of a liquid under pressure. The hydrometer is placed into the glass pressure tube. Sufficient sample is introduced through the valve to cause the hydrometer to rise half of the length of the tube. So the density can be read directly here. And the next test is the moisture content. So many methods have proven useful for this purpose. The Carl Fisher method has been accepted to a great extent here, and sometimes gas chromatography has also been used to determine the moisture content of the product. Identification of the propellant. So how to identify the propellant means by simply using a gas chromatography technique and infrared spectroscopy has been used to identify the propellants here, and also to indicate the proportion of each components in the blender. Okay, and next test is the aerosol valve discharge rate. So this is determined by taking an aerosol product of a known weight and discharging the content for a given period of time using a standard apparatus. By reweighing the container after time limit has expired, the change in the weight per dispersed is the discharge rate here, so which can be expressed as gram per seconds. And the next test is the spray pattern. So a method for comparing the spray pattern obtained from different batches of materials or through the use of the different valves is available. The method is based on the impingement of the spray on a piece of a paper that has been treated with a dye taxol mixture. So depending on the nature of the aerosol, oil soluble or water soluble dye is used here. The particles that strikes the paper causes the dye to go into the solution and to be absorbed on the paper. So this gives a record of the spray which can be used for comparison purpose here. To control the amount of the material coming into contact with the paper, so the paper is attached to a rotating disc and then has an adjustable slit here. Okay, this is how we will determine the spray patterns of the different valves of the aerosols. And the next test is doses with metered valves. Several points must be considered. Reproducibility of the doses each time the valve is depressed, and the amount of the medication actually received by the patient is needed to be determined. So reproducibility of the doses may be determined by simply assay techniques, so whereby 1 or 2 doses are dispersed into a solvent or onto the material that absorbs the active ingredients. So these solutions can be assayed and the amount of the active ingredient is determined here. And another method that can be used involves accurate weighing of the filled container followed by dispersing of the several doses. The container can be reweighed and the differences in the weight divided by the number of the doses dispersed gives the average dose here. So this must be then repeated and the results are compared to determine the doses of the product. And determination of the dose received by a patient is a rather difficult procedure here, since all of the materials dispersed is not carried to the respiratory tract. An artificial respiratory system has been developed and is satisfactory for this purpose. So and the next test is the net content determination. So several methods can be used to determine whether the sufficient product has been placed into the each container. The tiered cans that have been placed on the filled lines are reweighed and the differences in the weight is equal to the net content here. The other method is the destructive method. Under this, it consists of weighing a full container and then dispersing the content. The contents are then weighed with a previous being made for the amount retained in the container. Other modification consists of opening the container and removing as much of the product as possible. These tests are not indicated in determining the actual net weight of each container as related to the amount that can actually be dispensed. And the next test is the foam stability test. So here various methods have been suggested for the determination of the foam stability here. The life of a foam can ranges from few seconds to 1 hour or more depending upon the formulation. Some of the factors involved in controlling the stability of the foam is there. So what are those means? Visual evaluation, time for given mass to penetrate the foam, and the time for the given rod that is inserted into the foam to fall, and the use of rotational viscometers. So this is how we will determine the foam stability. Particle size determination. So many methods have been advanced for the measurements of the particle size of the aerosols. Among those that have been used to a great extent are the cascade impactor and the light scatter decay method. The cascade impactor operates on the principle that in a stream of a particle projected through a series of the nozzles and a glass slides at a high velocity, the larger particles becomes impacted first on the lower velocity stages and the smaller particles passes on and are collected at high velocity stages. A unit suitable for the analysis of the particles whose diameter ranges from 0.1 to 30 microns here. And then biological tests. The final phase involved in comprehensive research and development program for the pharmaceutical aerosols must involve the biological testing. A limited number of these tests have been used to evaluate the efficiency of many products, including various antibacterial agents. These tests are similar to tests performed on a non-aerosol pharmaceuticals. Biological testing of an aerosol product should include a consideration of therapeutic efficiency and toxicity. So we need to determine these two factors, that is therapeutic efficiency and the toxicity under this biological test. So how to determine this therapeutic efficiency means various testing procedures are available to determine the therapeutic efficiency of the aerosols. The doses of the product has to be determined for inhalation of aerosols and thus must be related to a particle size distribution. So topical preparations are applied to the test area in a usual manner and absorption of the therapeutic ingredients can be determined here. This is how we will determine the therapeutic activity of the drug. And then toxicity determination. The toxicity testing should include both topical and inhalation effects. Aerosols applied topically may be irritating to the affected area and may cause a chilling effect. The degree of chilling effect depends on the type and the amount of the propellant present here. So this is how we will determine the toxicity of the product. So all these tests are needed to be conducted for the evaluation of the aerosol products, that is flammability and combustibility tests, physiochemical tests, performance tests and biological tests are needed to be conducted for aerosol preparations. Thank you for watching this video. Please subscribe to our channel for more videos.